And that's from the forest, right? Or the
No,
forest
this is
end?
from, I swapped the sample. This is from Mike's, by the 60-inch.
Well, that's impressive to get a nematode. I didn't think we'd find any.
These people get really excited when they see a nematode. I hate to miss it.
You get a nematode, that means you have really good bacteria, because most of the aerobic nematodes that need oxygen can only eat aerobic bacteria. So you have to be growing a lot of oxygenated or really good bacteria. Your soil is aerated enough to grow that bacteria.
So what are some of these farmers in California that grow strawberries? They cover the whole field with a huge sheet of plastic and they put a gas in there to kill the nematodes.
Yeah, they have compacted the soil through years and years of tillage. Compaction on the soil is not directly causing problems in the soil. It's causing it indirectly, because once water infiltrates into the soil, it hits that compaction zone. And water doesn't move through two densities of soil very fast, so it's hung up there. So the water's hanging up at your plow pan layer whenever you till. That water hangs up there, and it only takes 10 minutes for the non-oxygenated bacteria, the anaerobic bacteria and fungi to take over. And again, it's not really them that cause the problem, it's what they produce. They produce acids and alcohols.
Your computer.
And that acid and alcohol is what destroys your root system. It what allows the root-feeding nematodes to proliferate.
So if the soil was healthier and you had nematodes, it wouldn't be the same problem then?
Oh yes, yeah. You want nematodes. You want there's a variety of different nematodes. There's predatory nematodes, and they eat all the other nematodes along with other organisms. There's bacterial feeding nematodes. There's fungal feeding nematodes. Those are all beneficial. And then there's the root feeding nematode. Now the bacteria and fungal feeding and predatory nematodes are in really good soil structure because they're large nematodes, so they need a big pore space to move through. If you till, you destroy the pore space, you make the soil less oxygenated, you have a high water table in the soil which decreases the amount of oxygen, and the root feeding nematodes don't need a lot of oxygen. And they're very small, so they can go through those pore spaces, and then they will attack your roots.
Okay, thank you.
So the best way to get rid of root-feeding nematodes is to do the soil health principles and reaggregate your soil, and never till if you can, or understand the consequences when you till.
Okay, so,
Just a minute, under a microscope, can you tell the difference between a root-feeding nematode and a good one?
Yes, it's crazy. You can see immediately, you know what you have after you do that. After maybe like 15 minutes of us showing you how to use the microscope, you will know the difference between a root-feeding nematode versus a bacterial-feeding nematode versus a fungal-feeding nematode. And if you have fantastic soil, you have to pat yourself on your back if you have a predatory nematode. The only way you're going to get a predatory nematode is if you have fantastic soil biology, because that's kind of like a tiger in the woods. You have to have this massive prey base for this large predator to eat. So for a root-feeding nematode, right away you look at its mouth, and if you just see a spear, you'll see this metallic-looking spear, and then you see at the base of the spear big knobs. Then you know you've got a root-feeding nematode. So the way that root-feeding nematode works is it goes up to your plant root and it shoots the spear into the plant root, and then it uses its mouth to go around the big knobs, and then it pries down to break open that plant cell, and then it latches on and then sucks out the juices that way. Now a fungal-feeding nematode has the spear, but it does not have the knobs at all, and you'll tell that immediately. And then the fungal-feeding nematode is a good 3 times bigger than the other one, because the root-feeding nematode can only be in your soil if your soil is compacted and there's not a lot of oxygen in it. So they function best in the conditions that are created by tillage. So if you have nematodes, you don't fix it with a nemicide, which kills all the nematodes and also the bacteria and fungi. You fix it by getting air back into your soil, by doing no-till cover crops, reduce and eliminate fertilizer, pesticides, and then most importantly, understand the consequences of one tillage event. or one application of fertilizer are pesticides, what that does. And then you can move forward instead of kind of digging yourself a deeper and deeper pit.
So this is going to be a fungi. Hopefully y'all can see it on the TV. So what we're looking for fungi when Michael and I are looking, we're trying to see if it has these what's called septae. So it's a crosshair, or it's a line that just goes across the fungi. This one does. So it's right there. And then there's one right there. That's probably just some biology beneath it. I think there's one right there. And then it's colored. So we just go by some factors if it's beneficial or not. So it's colored, it has a septate, and then you see these little bacteria, and then you try to line it up across. If it's 3 micrometers or larger, it's going to be beneficial. So this guy's pretty good. It's colored.
And this is out of the 60-inch.
This is out of Mike's
So
field.
that's progress.
Pat yourself on
And
the
then
back.
Because we did this last year and we didn't find.
And I was trying to get to that nematode so we could talk about the mouth parts, but then I saw this guy and had to stop on the fungi.
Well, when we did the samples, last year, I was on top of the hill up here, and we were finding a protozoan called a ciliate, and that protozoan is only found in low oxygen conditions. So that protozoan was there because of years of tillage. So normally you wouldn't think sandy soil would have a wetland or a really anaerobic microorganism, but because of all the tillage that was done in the past, that soil was essentially without oxygen for a long period of time. So then you end up with what is called a ciliate. I'm not sure where,
Number 20.
right here. So this ciliate only eats anaerobic bacteria or low oxygen bacteria that take over your soil. So if you see one ciliate in this slide, so if the one that Alicia's looking at, if we found one of these ciliates, and they're very diagnostic, you'll know right away when you see them, then you know something is bad happening. You know the oxygen concentration is decreasing in your soil.
What you got there?
Okay, that's the nematode that we found earlier. So for this one, it has these little spikes on the outside. It doesn't have that spear. Oh, it is alive still. I thought it was dead.
Yeah.
So it doesn't have the spear and it doesn't have the knobs. So we can rule out the root feeder.
What magnification are you at there?
We're at 400.
Okay.
So it is very long, so you can see its tail starts there, and then it's a larger size. Well, it's longer, so that could also possibly rule out the root feeder. But past that, I mean, it does kind of have these lips, but I'm not sure what these little spikes on the outside.
I think they gather the bacteria.
Is that a
Yeah.
bacterial feeder? The
Oh yes,
spikes
that's a
on the outside?
bacteria feeder.
Okay.
Yeah, for sure.
So if
moving.
you stretch that out in real life, how many millimeters is it?
Not even.
So if you look at these little, like the tiniest bacteria, that'd be a micrometer. So we're looking.
Millionth of a meter.
Yeah.
Thousands
Yeah.
of thousands.
It'd be very small.
A micrometer, that's a thousand.
That would be just one of these little guys.
Like a hundredth of a meter.
This is
That
way
thing.
bigger than the bacteria. So
A
I'm not
hundredth of a
These
meter.
are pretty long
How-
though.
yeah, that's-
They're not,
That's 10
It's a
millimeters.
Well, this would probably be
Yeah,
50
that'd be 10
micrometers in length, which is the diameter of your hair, or roughly of hair.
And you couldn't see this on the slide. So if I took this and just showed you the slide, you wouldn't be able to see it. We're at 400x.
So
A thousandth of a
One micron.
millimeter, okay.
Yeah.
That's a hell of a lot more than 1/1000th of a meter.
So like prior to like 1980, soil science knew of these organisms, and they're like, yeah, we don't know what they do. They just come back. They're really not important. But the biomass of nematodes in the Great Plains more than doubled the biomass of the bison that used to be here. So that the nematodes were doing something, and what they're doing is this is the nutrient cycling that's going on out here right now. So when this bacterial feeder eats 3 bacteria, because the bacteria is the most concentrated organism on the planet for nutrients, it has too much nitrogen for that nematode to take. So the nematode has to get rid of that nitrogen that's in the bacteria, and it releases it as waste. So it releases the nitrogen after 3 or 4 bacteria it eats, and it releases it in the soil as ammonium. And that ammonium will be converted into nitrate depending on the soil microbiology. So the soil microbiology in this field is dominated by bacteria, so that ammonium released by the nematode will be quickly converted into nitrate, and then it will be sucked up by your plant roots. So this is the way that the bags of fertilizer that are in your soil, which are the bacteria and fungi, get converted into soluble forms of nitrogen and are released by your plant roots, and then the plant roots take it up.
You talk about fungi in the soil, and soybeans and corn, we're applying fungicides now. Does it have any impact on the fungi in the soil?
Yes, it kills it all. And it also kills the bacteria, it kills the nematodes, it kills everything. So
And if it doesn't land on the soil, then what?
it will eventually hit the soil. So yeah, yeah. It's really something you want to think twice about. Now you do get yield. I know everyone tells me they get a yield bump, but the yield bump they're getting is from the mycorrhizal fungus and what we call saprophytic fungus. That was the fungus that we saw in there that was colored. That's a saprophytic fungus. That's a free-living fungus. Mycorrhizal fungus makes associations with plant roots. So all the corn, soybeans, pretty much all of our production plants make associations with mycorrhizal fungus.
So would you recommend on corn for tar spot not to put fungicide on it?
Yes, don't put fungicide on it. I'd leave it alone and get the biology back, because the reason why you have tar spot is because you have an anaerobic zone in your soil created by tillage or some kind of activity that's created some anaerobic conditions in your soil that allow that anaerobic fungus. So tar spot is an anaerobic fungus.
I don't think Mike had tar spot here last year or the year before. I didn't. It just came this year.
Yeah.
It wasn't in my soil.
Well, it comes in based on anaerobic conditions. So if you get the biology going in your soil by making it aerobic, you created the conditions where that tar spot can't
So how
live.
long will it take for it to do this?
Well,
'Cause
you,
I'm gonna lose half my crop next year or 2 years.
well, you can do it right now by making sure you're using multi-species cover crops, by doing a nutrient management plan where you're not putting on large amounts of inorganic nitrogen, to get down to a level where you're putting on, try to get under 100 units of N, by taking credit for existing crop that you have, especially soybeans. That's one thing that we're working with our producers right now to take that credit. For instance, if you took the credit for your soybean yield and then multiplied your amount of organic matter that's in your soil, so each percentage point of organic matter in your soil is roughly about 20 to 30 units of mineralizable N. So if you had like 3 units of N, you have maybe 60 units of mineralizable N available for your corn crop when you plant it. And then you take your soybean yield, which may be 40 bushels. You use 40 units of N for your corn plant. You add those two together and you're getting close to 100 units of N right off the top of the bat. So if you put on another 100 units of split applied nitrogen, that will get you to where you need to be. And reason why I'm talking about nitrogen is when you put large amounts of nitrogen, it causes your soil to go anaerobic as well, because you're growing so much bacteria. When you put on that nitrogen, it sucks up all the oxygen in the soil, and it will go anaerobic for a period of time. And that period of time is when all these anaerobic fungi, these disease fungi, like you're talking tar spot, some of the Rhizoctonia, Fusarium, all need that anaerobic conditions to grow and then to sporulate, to spread.
So these guys make me crazy. Because I get so lost, but every now and then Michael says something, I can jump on it, and he just said split apply nitrogen.
Yes.
And part of the strategy there is dribble on the nitrogen at the time the plant needs it. And Bob, the salesman, says that's one of the nice things about the wider rows is you have much more access to the field.
Yeah.
And so you can, and you're growing the biology between the rows, and you're giving yourself a place for the small equipment to go to dribble on nitrogen.
Yep. So you add all those together, all these anaerobic conditions that occur with applying nitrogen, or other things like tillage or compaction. They create the avenues for these diseases. Pretty much all of our diseases are these anaerobic conditions where you have low oxygen. I'm going to stop talking. I went crazy on fungi.
Do you think diversifying your rotations plus cover crops would even further help with that?
Oh yes, so that when you could get off the fertilizer and make sure you're rotating, then if you put soybeans that year, you don't put on any fertilizer. So there you don't have the issue with, there's a bunch of issues with fertilizer. One of them is creating anaerobic conditions. The other is fertilizers or salts, so they suck water away from anything that wants to grow in your soil, and the first thing they suck it away is the soil microbiology. So when we, like on our family farm, when we split applied nitrogen, we put on 50 units of liquid 28, and I took the sample before we did, and we had bacteria and fungi, and I took the sample after, and all the bacteria were in stasis. They weren't moving because all the water got sucked out of them. And the same thing for the fungi. So we got to be careful with fertilizer, and we have to be careful with doing monoculture. So doing that is a really good idea.
And
Mike, isn't that where banding fertilizer is more effective than broadcast?
Yes, banding is a much better deal, and then you don't put on as much, only where you need it.
Right.
Yeah.
So everything of Mike's this year was banded.
Oh, great.
So this was all 15-inch bands, everything of his. We as a company, we've been working with the new technology as far as your RTK guidance, and we've actually been getting pretty good luck doing 30-inch on-row banding. So we'll have a guy send us his planter lines he wants to use, and we can lay a band right back in it. And we've been seeing really good results with it.
That's fantastic. Yeah.
It's for anybody that has the technology already, it's really easy to work into. But yeah, we've had really good luck with it. That's we're kind of in our second year of broadly doing it, but
Yeah.
I guess I don't know what it would take to try to run it out to, a 60 or a 90 or a double to do it like that. I'm sure it could be set up, but
Yeah.
we see a huge response out of the banding.
Yeah, very good. Very good questions. Thank you. Alicia,
All right.
I'm not talking anymore.
anything else, Alicia?
No, we're out of time.
Well, any questions? I mean,
At what point does soil become dirt?
Well, I'll give you my thing, and then Alicia will. If it's just bacterially dominated, then it's pretty much dirt, But it's crazy. It's like if we took a sample of the dirt that's on the path there where nothing's growing, it's filled with bacteria. I mean, it's literally moving off the slide. It's kind of shocking. So bacteria come roaring back after any type of disturbance. But when they come roaring back, they're going to eat any carbon that's in that area, and they'll turn it into more bacterial biomass. So that's why people till. One of the reasons is they're cycling nutrients by having the bacteria eat the carbon in your soil. So I heard, Gabe Brown say that tillage is to the soil as fracking is to the petroleum industry. What it does is it allows you to mine your organic matter. And then he also went to say as fertilizer, and I'm not picking on fertilizer, but heavy use of fertilizer, but fertilizer is to your soil as fracking fluid is to the petroleum industry. It's essentially allowing you to burn the organic matter in your soil.
Yeah, so technically none of it's dirt. So
Yeah.
we do have life in it all. But if you're looking at diversity, then there is some that's basically no diversity, just bacteria. So it's awesome that we were able to find stuff in Mike's soil because this worked even better.
Well, we did some soil checking here last year when I got with Jacob over here, because he wasn't my supplier the last 20 years. He just came online, like you said, last year, pretty quick. And we had CSRs in the 80s up here. I've never seen that before. And it's going to be interesting to see what that next soil test is going to look like. But I think that no till it's starting to come back around. And I'm thanking God that I had the patience and the resources to stand it, because it was hard for me to get that out of my head not to till. This is how you're brought up. And I'm starting to see the benefits, and that's good. And we can kind of look around and see the wildlife that's hanging around here now that we haven't seen hanging around before. Like some of the little bluebirds and stuff, they're coming back. I mean, goldfinches hang out up here. We're seeing more bees and that type of thing, because we're not putting on the heavy duty stuff anymore like we were. I mean, I think Schaefer, you'll attest to this. When I first came to you, you said, Mike, I'm going to send you an agronomist out. So that's the day I reduced my expenses 40% because I found I wouldn't need fertilizer. I've been using the elevator for years to tell me what I need. And that's not a good idea. You need an agronomist to tell you what you need, because they're in the business to sell fertilizer. I paid for that lesson. But now that we're cutting back on that stuff, I'm in a nutrient reduction program via the USDA again. I got to say thank you to all of those folks, NRCS, the USDA. They've done a very good job of helping me get this place back in order. Had I not got involved with them, I'd be out of business. I would have just quit farming, maybe turn this into a development area up here, and the rest of it, I just have to let it go, because it's too much time and money and whatnot you have to invest to not make money.
Sounds like a factory manager company. So this is good. Thank you guys. You do make me crazy, but I love you dearly. I mean, it's just amazing. And Michael got me to get a microscope, and I've spent time at it. What's cool about this is it puts it on the computer screen. And I've never been able to manage the eyepieces very well, but it's really slick when you can capture it on your computer. You can screen capture it and all that. So if you're a little interested in it, it's a pretty interesting rabbit hole to go down, and it is a rabbit hole. So let's talk just a little bit about equipment, then we'll call it good. We'll call it a day. But I do want to, if you turn around here, look.